Okay, so part 2 of the bioinformatics session. We have, just to remind you where we're up to, this is your PCR product sequence. We've added the restriction digest in the lab and we've digested the DNA, your PCR product, and this is the wild type normal sequence, and it should have cut twice. Once there, once there. Using that information, you should be able to predict how big the bands are on the gel. There should be a little one here, a medium-sized one here, and a big one here. So that's what you would expect in wild-type DNA. Now what we need to do now is find out what the sequence is for the mutant DNA. So this is mutant BRAF. Okay, so it's going to be largely the same sequence. So I'm going to copy that and paste it there. And just make sure so we don't get confused. This is wild type. And this is mutant. So we know it's mutant, and we know it's mutant in the cell line A375, and that contains a mutation of BRAF that we are looking for. Now the mutation is known as V600E. Now that's useful information. That means that it's a valine at position 600 of the BRAF gene, which is mutated to an E, which is a glutamic acid, I think. So that's a mutation. What we need to do is just verify for ourselves that this is the correct sequence that covers amino acid 600, and we think that amino acid 600 should be around about there. Okay, so to verify where amino acid 600 is coded on this sequence, we're going to go back to NCBI, so www.ncbi.nlm. That brings up where we want to be. Under gene, I'm just going to go and search again for BRAF. There's the gene. Then scroll down to genomic regions and transcripts, reference sequence, and then I'm looking for, instead of here where we search for a genomic sequence, I'm going to search for an mRNA sequence, and this NM0034333.4 is the mRNA sequence of BRAF, or the coding sequence. So if I scroll down here, it tells you all sorts of interesting things about the gene, such as all the various different features. I'm just looking for the important bit. That's the amino acid sequence. Okay, somewhere in here it should tell you where the coding sequence starts, but that doesn't matter because we're going to scroll down and take this entire sequence. So that is effectively the mRNA coding sequence. So I'm going to copy that, open up a new window, and search for ORF Finder. Okay, ORF Finder in NCBI, that's the one. So what this is doing is screening a sequence and looking for possible coding regions where that DNA sensibly codes for amino acid. It looks for all 6 open reading frames in all 6 possible reading frames. So that means a coding sequence could start at, say, at the G there, or the T, or the following T, or the coding sequence could actually read backwards. So there are 6 possibilities. Either way, we're going to look for all of them. So that's the mRNA for BRAF. We've put it into ORF Finder, and ORF Finder has come up with this view. Okay, so that all of these are possible open reading frames from the sequence that we put in. So what it's done is identified possible open reading frames. This is one that's 766 amino acid. That's very promising, but all of these others are much shorter, so they're not likely to be proteins. So this long one at 766 amino acids is promising. So we can display the open reading frame as coding sequence and how it translates into amino acids, which we could then just copy out, put into our master Word document, which is what I've done here. Okay, so if this is right, we should be able to go down this sequence to get to amino acid 600. That should be roughly at base pair 1800. And each line of text here is 30 bases. So we want to be going down the appropriate number of bases until we get to position 1800. Okay, so what I've just been doing for a couple of minutes is counting down these rows of text. Each is 30 bases in length, and I'm down to here. And according to my calculations, this region here should be position 1800 on that sequence. I can assure you it is, because if you look at the sequence CAGTG, that is the recognition sequence of our enzyme, and that there is an amino acid V for valine. So what's going to happen in the mutation is that one of these bases somewhere in this recognition sequence is going to be mutated, so that this valine now cleaves for a glutamic acid, and the restriction enzyme will no longer cut. So I'm happy that the sequence of our primers is recognizing a bit of the DNA that codes for the region around valine 600. We've got primers that work in the right place, which is something that we have now verified. So that means that we've now virtually completed part 3. All we have to do now is search for a scientific paper that's got the V600E mutation in it, and hopefully it will contain some sequence that resembles this here, and we'll be able to work out exactly which base is mutated with the V600E mutation, which is what we're looking for. So all I'm going to do is go to a new page, type in V600E BRAF, just see what comes up. Okay, so here's a paper that describes it, just happens to be the top hit, and I'm going to go through that paper and see if there's any sequence information. Okay, so that paper that I clicked on didn't have any sequence information, but this one does. So all I've done, same search, and I've clicked on images, so that should say V600E BRAF, and I just clicked on images, and I came up with this little bit of sequencing that I recognise. This is from the paper that is, the hyperlink is on your text file. It's a journal in Journal of Clinical Pathology, and you can get the full text of that paper. It's open access. So there you go, the paper by Agami et al. So this paper has characterized and sequenced the BRAF sequence, and they are telling us exactly what the sequence is that we should be looking for. Get rid of that advert. Okay, sorry about that. Okay, so what we can see from this is they say the wild type sequence goes CACTGT and the mutated is CTCTGT. Okay, now this might be a bit confusing. What they're showing is the antisense strand to your PCR product. So all you need to do is write this out backwards and reverse antisense it. Okay, so let's go back to the document with our PCR products. I'm just going to scroll back up. Okay, so we're looking for a sequence that goes, I think it said TTT, CAC, TGT. Well, if I reverse antisense from here, TTT, CAC, I'll start again. It goes TTT, CAC, TGT. So I'm reading back those 9 bases backwards and antisensing them, and those are the bases that are shown here. Okay, now if that's the mutant, they are saying that the A is the wild type, okay, and that's mutated to a T. So what we need to do is be able to swap those bases round. So having done the little conversion in my head, I think that that T there, according to their sequencing data, becomes an A, which I'll just put in a different colour so there's no arguing about it. Okay, so their sequence went TTT, CAC, TGT, and the mutant went TTT, GTG, TGT, I think. Yep. So all I've done is introduce a mutation that matches what they've done. Now some other papers might have got the sense strand. That'll make your life a bit easier. Anyway, all we've done now is what I've done is introduced what should be the mutation that is well characterized in the V600E mutation in melanoma. So next step, this is the mutant sequence. So if we copy this sequence out and go back to restriction mapper, I'll just find it again, restriction mapper. Okay, paste the sequence in, map sites, and what we now see is that the TSPR1 site only has a single cut site at position 211. Okay, compare that to the previous one where we have two cut sites, 211 and 124. Okay, armed with that information, you should be able to figure out what on earth went on with the gels, and that's based on the data that I sent to you. Okay, so the point I did there was actually everything in point 4, which is where I've identified the possible restriction sites. I've used that information to put them into my DNA sequence, put them into restriction mapper, and what we have now got is two different documents with different cut sites. So that is a wild type, that is the mutant, because what I've done is changed that T to an A, and that is the mutation that we've verified from the paper by Agami et al. That is what happens in the V600E mutation. We've also verified that this bit of DNA codes for codon 600, which is correct. So therefore you have all the information that you need to be able to predict the genotype of the A375 DNA and be absolutely sure of what the banding pattern should be. Now just be aware, when you're trying to work out the banding pattern on your gel, you've got the sizes. So for example, we're expecting that to cut there, we're expecting that to cut there, and that gives you 3 bits of DNA. You should have got 3 bands on the gel. Whereas on this one, this site has been destroyed, this one will cut, so you've got 2 bits of DNA. Okay, you've got to rationalize that information with the information on the gel. Okay, and that is the main part of the interpreting the gel. What's also interesting is this mutation in A375. You've got to figure out if it's homozygous or heterozygous. That should be obvious to you. But I want you to present information on the gel for all possible genotypes. Okay, so go through this before the session, and then on Wednesday there'll be me and Lucy who'll be taking you through all of this, and we'll probably start from scratch for everybody, but at least when we get to the bit where you're stuck, we'll be there to try and sort you out. Okay, good luck.